Somewhere around here, I think we have the budding in the corner that you're going to be seeing real soon. I highlighted it. You're going to be seeing it really, really soon. It doesn't seem to be appearing, but in the corner, there you go. Inside of the corner, you can see the yeast buds, how it buds. The way I've explained it in the slides is the same principle live in a microscopy. It's super impressive the way it happens.
Hey guys, just to let you know that I have a Patreon.
Nice.
If you are interested in supporting me, head on over to patreon.com and subscribe for some exclusive content.
Hey, welcome ladies and gentlemen to this amazing stream of science, obviously. Because I was working so hard on this for the past month, and I had to do it. We're going to be learning about Mycetes today, about these guys. The reason why I wanted to do like a Mycetes video is because there's not really that much information about fungus. Mycetes, fungus, however you want to call it. The majority of people call it fungus, but the way I was taught was with the word Mycetes. Which is the same thing, basically, technically. So without further ado, let's get straight into it with the characteristic of Mycetes. Like I said, there's not really that much information about eukaryotic infectious agents, and especially about Mycetes, about fungus. So I thought it may be like a little bit fun to like maybe teach you about eukaryotic agents, infectious agents such as these guys, because as you can see, right, if I can just like activate my pencil, as you can see over here, these guys are in the Eukarya kingdom. They're in basically Eukarya. If you have done your science classes or your biology classes, you will know that in taxonomy, it means that it's our cells. Our animal cells, human cells. Yes, indeed, fungus derive. Fungus are not considered plants, technically. They're considered animals. Well, not. How can I put this? Because animal cells derived from these guys. So these guys are part in that lineage. They're not going towards the plant lineage because they don't have. They don't have the same characteristics as plants, and that's why. So if we go down the taxonomy tree, this is a more simplified taxonomy tree. We got the Mycetes over here, and technically you could add like a third one over here and call it like what's the name for fungus in like not the molds, not the yeasts, but the mushrooms. Yeah, you could technically add here like mushrooms, but for the sake of this video, usually mushrooms, they're more poisonous than anything else, but the main infectious agents that you're gonna find are either molds or yeasts, and those are a little bit what you're going to find in infectious agents when you get infected by these things. Yeasts, their main characteristics is that they're unicellular. Molds are multicellular, and we're going to see why. And if we go into like really quickly into the characteristics, we saw they're eukaryotic. Oh, this is very interesting. Opportunistic. They're considered opportunistic, the majority, because they don't infect like a regular pathogenic agent. They'll usually infect when you have a decrease, for example, a decreased immune system, most notably with children because they have a weaker immune system growing up, or when you're older, when you get older, you have like your immunity system decreases, or if you have like, for example, you're taking corticosteroids, that could also influence a part of the opportunistic agent part of the fungus to start infecting, because corticosteroids lowers your immune system, right? So that's how they're considered. If they have also as well, if they have the proper environment, and inside of the body, it is a really good proper environment because they have access to humidity. Because one of the characteristic is that, if I'm not mistaken, the molds specifically over here, they can live. If there is enough humidity, they grow exponentially. And the way that they grow is really interesting. If you've seen a mushroom in the wild, in a forest, you're going to see the tip of the head, right? You're going to see like, if I can just draw it over here, you're just going to see like this over here, like that. You're just going to see here. What you're not going to see is that this is also, and this is a small mushroom because it's going to go like really far. It really goes like really far. They grow not an exponential rate, but they cover a quick amount of surface when fungus grows. That's also really interesting part of fungus. Mycosal infections, they're divided into 3 classes. I'm not sure how teachers usually teach, but the way I was taught is that you got 3 of them. So you got the first one being cutaneous, so the exterior part of the skin, subcutaneous, inside of the skin, and finally systemic, where it just goes into multiple organs. And that's the way I was kind of taught at school about pathogenic fungus infections. Moving on, as I said, they cover a great amount of surface when they grow. As you can see, like I said, this is probably the tip, but at the bottom you're gonna have like these like really kilometers of like fungus that grow, having only one mushroom tip, you know, which is really interesting. Unicellular, multicellular, that's fine, that was said. Heterotrophs, that's actually a really interesting part, meaning that they consume food. Like I said, they don't share the same characteristic as plants. They will actually consume their food. And that's really interesting because, you know, in the wild, with like a mushroom over here, with like the mushroom, they're going to actually encrust themselves. They're going to go inside of the plant cells and start consuming their the food that they make from photosynthesis. So that's super interesting. Same thing actually happens here, or for example as well, like excrement. They consume just about anything that we as humans can't, you know? Mostly like excrements, waste, biological waste. They're going to consume it, and they're going to spit it out back as like organic compounds, you know, which is super, super really interesting. And this is what the one I was trying to see. So for molds, they can tolerate dehydration. Yeasts greatly need water, but in both cases, molds and yeast, if they have a very humid environment, they grow exponentially and cover a great amount of surface. So it's actually for both. Molds are strictly aerobic, meaning that they need oxygen. And yeasts have, they don't necessarily need oxygen to grow. They can grow as well. But yeah, it's as needed. All right, now that we got the characteristic out of the way, I hope that was really helpful. We will now be going on to just to know about the fungal anatomy, the fungal cell anatomy. General similarities, like I said, we said it before a few seconds ago. They're eukaryotic agents, so they have the typical components. Oh, whoops. Oh my goodness. Oh my god. I screwed it up, didn't I? I screwed it up, didn't I? Okay. I screwed it up, didn't I? That's what I was looking for. I don't know what the hell I did, but whatever. So, you're obviously going to find mitochondria, centrioles, peroxisomes, rough endoplasmic ribosomes, nucleus, everything that basically, if you had your basic biology class, you're going to see that they have every single component. What differentiates, if I'm not mistaken, they have a vacuole. Interestingly enough, just like plants, even though we said that they don't share the same functional characteristics as plants, they do have a vacuole. They have lipid granules or lipid bodies. They have on top of their where is it? Their cell, their phospholipid bilayer. Huh? Didn't I put it? Golgi apparatus. Huh, I did not place that. So yeah, basically, they have this cell, it's not a cell wall. I forgot what it was. But it's the phospholipid bilayer. It's the, it's the phospholipid, bilayer. That they have, that also we have, you know? But they, on top of that, have a cell wall, which is here, which protects the fungus cell from stuff like phagocytosis. It makes it a lot harder, you know, and it protects against it for stuff like that. And then finally, you have the bud scar over here, because one of the characteristics of yeast is that the way that they divide is by budding, meaning that compared to molds, which are over here, yeasts, let's say that this is a yeast cell. What's going to happen is that the yeast cell, when it's going to divide, it's going to like divide from inside of itself, and it's going to create another yeast cell that's going to grow like that. And this part over here is called the budding mechanic, if you will. And if you're thinking also about the budding, the way that some viruses bud out of the cell that it infects, it's kind of similar. They're both like taking a part of because budding, the definition of the word budding, is that it's gonna takes a part of the mother cell with it, right? And this is where you get the bud scar. Although that's where the similarities stop between the virus and the fungus cell. The fungus cell will actually have this bud scar once it detaches from the mother cell that it divided from. Finally, we go on to molds. Molds, as you can see, they grow in a sort of what we call hives. This over here is called hives, or hyphae, actually, if I should say, hyphae, for plural. They're gonna grow like this, one on top of the other, you know, and it's really different from the yeast cells. Very, very, very different. But again, you're all gonna find the same similarities in the yeast cell as in the mold cell. Mitochondria, nucleus, smooth endoplasmic reticulum, etc., etc. Lipid granule, vacuole. But the only difference from the yeast cell is that you're gonna have a septum. The septum is like this separation that separates, for example, this cell from this cell. And like I said, one of the characteristics of molds versus the yeast is that the molds are multicellular organisms versus the unicellular from the yeast, even though that sometimes they don't bud out. The yeast cells are not going to bud out completely, and they're going to have like, they're just going to stay budded together. If you have multiple, and you're going to see in the video, because I have also a microscopy video But let's not worry about that for now. Let's go straight into cell division, and because I wanted to also showcase what cell division is for the fungus, like a general fungal development cycle. Mainly because there's two differences. What's really interesting is that inside of the fungus, you can either have haploid cells, meaning that you have gametes that are gonna fuse together to create a diploid, or a zygote, if you wish to call it that. Or you could also have the zygote go through mitosis, as you can see here in this cycle over here. You can have them go through mitosis as well, so they can be both. It's super interesting. So if I should start, you have the haploid mycelia over here. Eventually, they're going to meet with a male and female. They're going to meet, a male is going to meet a female. And they're both going to, like, integrate the same cell, you know? They're going to become dikaryotic. And as you can see, this is like the tip of the mushroom that grows, and I didn't really want to just go specifically into like yeast fungus, so I tried to like condense everything together. So if I have like a mycologist, a professional mycologist, and I'm making mistakes right now, please do not hesitate to correct this slide specifically, or any slide whatsoever. I will do the changes as well. If you tell me that I'm making a mistake or whatsoever, I'm really open to constructive criticism. What happens here is that inside of these like gills, you're gonna find these oh my god. There we go. It takes a long time to load. But these basida, I don't know what they're called. Anyways, eventually these guys fuse. They fuse inside to form a zygote. These zygote become I have to let it load. These zygote become diploids, and you have to let it load again. There you go. They become diploidic, and they go through meiosis, eventually creating spores inside of 4 haploid nuclei. These spores eventually disperse and germinate. They go into the ground, and they have 2 separate pathways where they begin growing as a yeast. And this yeast cell, this mold-ish cell, or yeast, however you want to call it, will go either through the mitosis cycle, or they're just going to restart the same haploid cycle over and over again, you know? If they go through mitosis, I'll just go really quickly. For those who don't know what it is, it's basically asexual reproduction, where you have the metaphase, then, the prometaphase, then the metaphase, then the anaphase, telophase, and etc. And what's really interesting is that here you're going to start to have a new cell that grows out of the old one, eventually forming a mycelium. This actually is a smaller, condensed version of the mycelium. Usually it's a lot bigger than that, but this is just to show you an example of what a mycelium actually is. And then finally, once the mycelium has been created, it eventually sporulates and then germinates again forming mold cells, yeast cells, starting the whole sexual reproduction cycle all over again, you know? So that's basically what this whole slide is. It's just to give you a little preview of what fungal development cycle actually is. In any case, moving on now. We go on into the mycosal infections, and hopefully I can I can probably not lose a few folks over here. But in any case, we're going to start off with the cutaneous ones. I picked a few important ones that you may see inside of your professional practice. And I regrouped the cutaneous and subcutaneous together because they mostly do the same thing. The most common one, even though that the first one is going to be candidosis, and I'm putting a little parentheses on candidosis because it could also be systemic. But in general, candidosis or candidea or candidosis, that's the way I learned it. You're gonna find that it's really prominent in cutaneous infections. Most notably, out the top of my head, is athlete's foot. Or, out the top of my head again, when you take puffers, when you take your orange puffers specifically for those people who have asthma, for example, or for the people that have, who have been prescribed an orange pump with their COPD, because that is possible. Often we're going to tell everybody to who have these pumps to wash their mouth, you know, after using their orange pump. And that's the reason, because they're corticosteroids and you're prone to having the candidosa grow on your tongue. Labial candidosa, you know? So that's another example. I'm trying to make it unload. In any case, general symptoms really vary from eccentric side, and what's really interesting is that candidosis, candida, sorry, that's the name, the pathogenic agent is candida. Candida is already prominent in the normal human microbiota, which is really interesting to know, that it just stays there, but it's suppressed. It just stays there silently. Like I said, it's really every fungus in the majority is opportunistic, meaning that it just stays there, cool on the side, not bothering anybody. But if there's a weakness somewhere, that's when it starts attacking. You know, that's super amazing. If there's not really an incubation period, and then the treatments include antifungal agents such as echinocandin, fluconazole, amphotericin B via IV tablets, cream or ointment. Cream or ointment is really going to be for the skin. IV is going to be really for the systemic when it starts affecting the various organs. I'll just go a little bit down, and as you can see, the most prominent what I had, you can also have it inside of the nails, and inside of the nails, I guess that you could say that it's subcutaneous. But as you can see here on the foot, he has it. On the skin, he has it. On the foot, like I said, it's most commonly is going to be athlete's foot. You know, if you don't wash properly your feet and you don't, after you wash, you pass to remove the water between the toes, you're prone to having that, you know, because it's a really good humid place for fungal growth. You know, if you have a cut, it even gets worse. He gets into your subcutaneous region and starts affecting your or the inside layer of the skin, you know. So it is what it is. You could also have it at the sexual organs, you know. It could be vaginal or penile infections, you know. So it is really prominent when you get these the acandidosis infection. The reason why I put it in cutaneous is because it's the most common one. But like I said, you could also have it subcutaneously, and in parentheses, you could also have it systemically, as you can see here when it infects the various organ. Moving on to the lower one is the Sporotrytosis, or Sporotrycosis. General symptoms are going to be ulcers that you're going to have, and also lymphatic node ulcers. So you're going to have like lymphatic node growth, and you'll see it prominently as well. Usually it's contact with soil, and the incubation is 1 to 12 weeks. And the treatments are antifungal agents such as isoconazoles via tablets. That is the most effective treatments. Again, the why I don't have any infographics is because it generally affects the subcutaneous part, so like the skin, again, of the affected region. And lymphatic is that it infects also various organs, the lymphatic lymph nodes, you know? So that's probably one of the main reasons why I didn't put any infographics for that one, because it's kind of the same thing, you know. It is still like a yeast infection, like this yeast over here, but that's pretty much where it stops. Moving on to the systemic one, probably one of the most interesting and most dangerous one is Cryptococcosis. The pathogenic agent is the Cryptococcus, mainly because of its shape. And general symptoms include pneumonia, and if untreated or not treated properly, it can complicate into meningitis. Spore inhalation is the entry type. As you can see here, you got these spores that penetrate inside of the system. They diffuse into the systemic circulation, into the pulmonary circulation, into the blood vessels, eventually entering the circulation of the blood, crossing over into the blood-brain barrier, and causing what we call cryptococcal meningitis, which is really, really dangerous, because then you have to find stronger drugs that can go through that can pass through the blood-brain barrier to fight off these yeast cells. Treatments include Flucitocin, amphotericin B, sorry about that, blee. Fluconazole via IV or tablets. If I'm not mistaken, the Flucitocin is the one that can be given through amphotericin B is the one that can be given through nebulization to go directly into the lungs. The Flucitocin is I think it's the one that can pass through the blood-brain barrier. I think. I'm not mistaken, but correct me on that one if I'm wrong. These are the treatments. Moving on. Then we have Pneumocystis Pneumonia. General symptoms, again, it starts affecting the lungs, so general symptoms is pneumonia. Generally asymptomatics in adults, but prominent with immunodeficient individuals. So, for example, like I said, at the top of my head, children, the elderly, or people taking immunosuppressants such as corticosteroids. Very important to note. Again, entry type is the spore inhalation, and again, that's the reason why there's no infographics for that one, because it's spore inhalations. You can just look at this one over here with the spores being transferred into the systemic circulation, you know? Incubation period 4 to 8 weeks. And then finally is antifungal agents again, such as trimethoprim, sulfamethoxazole, the TMP type or the SMX type via IV or tablets. Moving on to the other fungal pathogen, which is Asper Juliosis. This one is really interesting because it has two forms, if I'm not mistaken. General symptoms include allergic reactions and bronchitis, inflammations of the lungs. Again, spore inhalation, the incubation period is variable, and the treatments are voriconasal, isovuconasal via IV tablets or suspension. And it's really interesting because, let me just make sure that it's this one actually, and not the one below. Oh, it's actually the one below, actually, I think. Okay, yeah, it is the one below. No, this one is nothing really special, but I think this one is that it creates fungal growth, but this one is more like a mold cell pathogen. It is a mold cell pathogen. I think it's the only one on the list that I had in the most common ones that affect the tissues. It starts affecting the tissues inside the lungs specifically. And as you can see, it starts growing in between it. And it starts stealing the nutrients from the from the epithelial cells, which is crazy to think about. That's why they're called opportunistic pathogenic agents. If you have the chance, if you have immunosuppressive stuff inside your body, basically. Finally, there's the last one, is Blastomycosis. This one was the one that, interestingly, I wanted to talk about because the way that it infects, it all begins... the spores begin into mold shape. Where you inhale the pathogens, it goes into the pulmonary system. And the body temperature actually modifies its growth into yeast form. That's crazy. That's crazy, bro. That's super crazy. It modifies the mold, which has this distinct shape, so it doesn't grow as a mold, but with the body temperature, it transforms it into yeasts. These yeasts eventually make it into the blood circulation, and they get phagocytosed by the white blood cells. But it doesn't matter because they evade the phagocytosis because of their thick cell walls. It's really crazy, wow, how that happens. General symptoms include pneumonia, ulcers, and cutaneous abscess or abscess. Spore inhalation, as I said, 3 to 15 weeks, and the only super effective antifungal agents are itraconazole, amphotericin B, via IV or tablets. So you're gonna notice that I've been talking a lot about these antifungal medication. And you probably have no idea what these do, but do not worry because I got you, fam. Because the next slide is going to be actually the antifungal medication mechanisms of actions. Starting off with the most common used ones, and this is where I'm actually gonna start by going a little bit deeper inside of the cell biology of a fungal cell. We saw in I think it was the second slide, about the anatomy of the fungal cell. We saw it a little bit. And you're going to see, this was the word I was looking for, the plasma membrane, that I couldn't just remember. So the plasma membrane, as you are going to see, is made up of all of these. So here you got, this part over here is your phospholipid bilayer. In between, you have a protein called ergosterol that is the same thing as, it's a derivative of cholesterol, but it's the main component of what keeps the phospholipid bilayer together. You know, ours is the cholesterol that binds and keeps everything together. In here, it's called ergosterol. That's the main type of protein. You're also going to find chitin in the plasma membrane on top of the phospholipid bilayer. Basically, the manoproteins, the glucans, and the chitins make up the cell wall. So this all the way over here is the cell wall, basically, made up of manoproteins, which are these, glucans, and then finally chitin. What interests us in our first medication, which is amphotericin / nystatin, is basically to affect the ergosterol, and you're going to see why. As you can see, you've probably seen it by now. This is amphotericin B. This is the main molecule of amphotericin B. Nystatin as well. It does the same effect, actually. The reason why I just put the amphotericin B molecule is because, I mean, if you people want me to put I'll also add the nystatin molecule, I will. But the reason why I didn't add the nystatin molecule is because it has the same composition. There's just like small molecular differences. It's really small, but it's essentially the same molecule. It has the same shape. I think it's probably in this part that you're gonna see like really small differences. But aside from that, it's basically the same molecule, and it does the same effect as amphotericin B. So that's why I didn't put it. But nystatin is going to do the same effect. It's basically going to create pores in the ergosterol, making it that the ions will leak out of the cell. And if ions leak out of the cell, it can't regulate its functions properly, leading eventually to cell death. You know, it leads to cell death. It leads to cell death, you know? Unfortunately, there are some side effects that we need to talk about, because if you take it in huge quantity, because even though that ergosterol is not prominent in human cells, taking it in huge quantities will affect the cholesterol component of certain epithelial cells, most notably the renal epithelial cells and the vascular epithelial cells inside of the kidneys, in the renal region. And the way it does that is the same thing, is that this is our cholesterol, and it's going to create pores inside of the cholesterol, destroying the cholesterol component, causing ion leakage, basically. It's going to cause that. So be careful with this. Usually creams, because of amphotericin B or nystatin, as creams or tablets are not that bad. But taking it in huge quantities in tablet form can be detrimental to, depending on the treatment that you want to do. Moving on to azoles. If we go on into azoles or allylamins, basically it's the same type of mechanism for both azoles or allylamins. It's the same mechanism of actions. It's just that the azoles, if I'm not mistaken, are going to affect the this enzyme over here. The allylamins are going to affect this enzyme over here. So what happens basically is that, again, we're going to try to affect the ergosterol component of the plasma membrane. And the way we're going to do that is by inhibiting the effects of this specific enzyme. If we start first with the squalene epoxidase, its only goal is to transform the squalene component, which you can see here, into lanosterol. The way you do that is with your alley lamins, for example. Those include naphthafines, terbinafines, or butenafines. They're going to inhibit specifically this enzyme over here, making it unable to convert squalene into lanosterol. Or with your azoles, most commonly used, for example, include clotrimazole, fluconazole, itraconazole, myconazole, ketoconazole, and voriconazole. Mainly the first two, clotrimazole and fluconazole, are the main ones that you're going to see for like skin infections, for example. Fluconazole, for example, the commercial name for it is Canesten. Canesten is the main one usually used for vaginitis. This one's gonna convert. It's fluconazole is the main one used for the vaginitis infection of yeasts. And basically, same thing. It's gonna affect a specific enzyme. This enzyme is your 14A demethylase, and again, this enzyme specifically will take the lanosterol component once it gets converted from squalene into lanosterol, and its only goal is to convert this into ergosterol. But if you inhibit it, will you be able to make ergosterol? No. Will you be able to have your phospholipid bilayer be sturdy and functional? No. Will you have ion leakage? Perhaps, yes. Will you have a stable plasma membrane? No, not at all. You're going to lead to cell lysis, leading to cell death. Bingo. We got that out of the way. Moving on to echinocandins. Echinocandins will affect another component that I did not talk about in the first one when I went a little bit more deeper into the plasma membranes, but we're still affecting the plasma membrane here. We're still inside of the plasma membrane. We're going to affect another component called the it's an enzyme over here. It's called the beta-1,3 glucan synthase, which is also responsible for the cell wall production. Echinocandins include Anidula fungens, capsofungins, or fungins. I hope I'm saying this right. Hopefully. Correct me if I'm wrong. These guys are just going to inhibit the functionality of this enzyme, of this protein enzyme that is in between the phospholipid bilayer. And if you're inhibiting the functionality of this, leading to osmotic instability, is it going to be able to complete its function? No. Is it going to lead to cell lysis? Yes. Yes, it's going to lead to cell lysis. And it's super effective against the Candida species and the Aspergillus species. So keep that in mind. Finally, we're going to move on to the ones... now we talked mainly about the ones affecting the plasma membrane. Now we're going to go into the ones that I found interesting affecting the inside of the cell. Mainly the grisofulvin. The grisofulvin actually is... it's actually really interesting, its mechanism of action. They don't know how or why it functions. It's idiopathic, meaning that we don't know why it works so well that it does. But basically, this molecule over here, which is the grisofulvin, is gonna go inside of the yeast cell or the fungal cell. It's gonna inhibit mitosis. It's gonna literally inhibit mitosis. It's super cool what it does. It's this Grisofulvin is going to bind on to all of these microtubules that get synthesized during the mitosis part, and they're going to attach onto it. They're going to latch onto it, and they're going to inhibit them latching onto the chromosome. If they inhibit the chromosomes, the microtubules latching onto the chromosomes, are they going to be able to properly divide into a new cell over here? No, they're not. It's going to slow down cellular division. It's going to slow down the mitosis process, leading to fewer and fewer multiplication of the yeast cell. It's super interesting. It slows it down, and with that, you can add on top of it other medication. For example, echinocandins, azoles, allylamines, or nystatin. You can add that on top of that because it's really good at inhibiting this, you know, and at getting rid of the yeast pathogen. Finally, we have Flucitocin. This one's also really super interesting. This is the molecule. It's Flucitocin. It's going to go inside of the fungal cell. It goes through a few conversions. For example, cytosine deaminase is an enzyme that is present inside of the fungal cell that is going to convert it into 5-fluorouracil. Then it's going to get converted. Once it gets converted into this, it's going to convert itself into 5-F-dump. I'm not going to name this whole name. I'm sorry about that. I'm not going to name it all in its complete form. I've already named this, but it is what it is. And then this specific molecular component is going to inhibit this enzyme, which is the thymidylate synthase, which takes the dump molecule. Usually it takes the dump molecule, converts it into a DTMP, which is usually DTMP favors DNA synthesis. But if you inhibit this enzyme, is it gonna be able to make more DTMP? No. Is it gonna be able to promote and increase DNA synthesis? No. It's gonna decrease it, actually. And if you decrease DNA synthesis, is it gonna be able to divide? No, you're gonna inhibit mitosis inside of the fungal cells. It's super impressive the way that it works, man. Man, this is super amazing. Learning about all this was like super amazing. It's really incredible. Aside from that, finally, we're going to be moving on to the references. These are all the references that I used. Thank you very much for the literature that I've used. I had to work a lot hard to get necessary informations to be able to like put everything into an hour. Now, finally, we get on to the main event. We're gonna get on to the microscopy video that I made, and it's only one microscopy video. And it's basically fungal cells, most specifically yeasts. The only one that I managed to take out from this was the yeast cell from cheese, from moldy cheese. Yeah, the blue cheese that some people like to eat and that's super expensive. I decided to take a sample from that, basically. And I decided to see if we could find some yeast cells to look at. And as you can see already at 40x, you could already start seeing the yeast cells inside of the microscope. It's super cool the way it does that. Let me just see if I can, there you go. I can like make it a little bit bigger. And this is 400 magnification, and you can see the individual yeast cells, as you can see. Somewhere around here, I think we have the budding in the corner that you're going to be seeing real soon. I highlighted it. You're going to be seeing it really, really soon. It doesn't seem to be appearing, but in the corner, there you go. Inside of the corner, you can see the yeast buds, how it buds. The way I've explained it in the slides is the same principle live in a microscopy. It's super impressive the way it happens. Moving on, you also have the only component that you can see is just the cell wall, basically. You can't really see inside of the yeast cell, which I find really interesting. I find it really interesting you can't see inside of the yeast cell like you can see as other, like the plasma membrane. You can't see it because it's covered by the cell wall. And that's what you're mainly seeing inside of these yeast cells. You know, that's the only component that you can see, you know? If we move on, sorry, my ears are super itchy. My ears are really super itchy. Moving on, if I can just like move on, yeah, it just stayed over here. Yeah, I just really like stayed on the C cell on the C cell wall. I tried highlighting it because this is the only thing that you can see. If I can just like, I'm just gonna fast forward a little bit because it really just stayed here. Oh, this is really super interesting. I'm not sure if this was an actual cell that was moving or if it was moving with the current of the water, because the way I did my sample, my sample to look under the microscope, I put a drop of water on top of the sample of the mold, of the cheese mold that I used. And I saw this cell moving, and I thought it was like super interesting the way that it moved. So I'm not sure if like a mycologist or somebody who studied biology could like properly give me some insight on if this cell is actually moving, because it's super interesting. You know, you're seeing it here in slow motion at actual speed, moving. But if I fast forward like this, you could actually see it moving. And then it just kind of disappears with the others. But look how big it is compared to the other ones. It's super interesting to see. We move on to another angle that I tried looking at. I tried to this one I tried to see more on the darkness part of the darker parts. You're gonna see a little bit. Yeah, I'm changing the magnification of it. As you can see, I tried to look at the darker parts to maybe see if I can like find something. There's not really that much to look at. It's super hard. I had to like add filters inside of this like microscopy video, so that you guys could see a lot better. I had to modify a few of the settings because it was super dark. It was super dark because this is like literally the blue part is a bunch of fungal cells together, and this is what you're seeing. It was super dark, so I had to increase like the luminosity and the contrast of it as well. You know, this is what you're literally seeing at 400 times magnification. You know, I'm gonna fast forward a little bit. This is what you're kind of seeing inside of the dark part. I managed to find a lighter part of it, but this is what it is, you know? If I think at some point I do decide to like look a little bit, not further, but like deeper or a little bit up, and I'm just gonna fast forward a little bit. I think it's here, but yeah. Oh, there you go. Yeah, I'm gonna leave it here. Yeah. I decide to look a little bit deeper or up. I'm not sure, but I eventually find another like yeast budding, like right here. It wasn't clear, but you could definitely make it out. Yeah, I think I was zooming out. But yeah, zooming in, you can see here clearly. I managed to find other cells that were budding. Oh, I'm just gonna pause here. I'm not sure if you guys are seeing the long filaments. I should have probably highlighted them, but maybe somebody could like correct me. But I think those are hyphae. They're hyphs. They're pseudo-hyphs maybe, but they look like hyphs, those filaments. So maybe I could get like a correction on the video if I have biologists or mycologists that are watching this, you know, who could probably help me out here. I don't know what it was. I didn't want to go into precision, but I did want to mention it, you know. I did want to give it a mention. Then here it's a bunch of darkness. I tried to look for more interesting stuff, but I couldn't. Oh, this is actually really interesting. It's a bunch of fungal cells together, you know. That's pretty much it. Then we move on to another part, and this was mostly the water pushing the mycosal cells together. And I kind of found an interesting part here. As you can see, all of the yeast cells or the fungal cells moving in one direction with the water. I think I zoomed in at this point. At some point, I wanted to find a good spot to zoom in. I think I'm gonna, yeah. Give me a second. I think there you go. This is where I zoom in. At this spot over here. Yeah, because I found it interesting of the fungus of the fungal cells just moving into one single flow. I went into 40x, but then I tried finding my oh my god, you can see them so bunched up. There's millions, maybe even billions of them, all bunched up together. That's super crazy, man. That's really super crazy, bro. It's incredible. It's super incredible. Ah, there you go. Now you see them alone. They're really big. I changed to 400x magnification, and I tried to see them like one by one. Honestly, at this point, I'm not sure if I'm looking at like different sizes of fungal cells, but maybe, again, mycologists or biologists could help me out here. But I just found it super interesting that there's like actual different sizes of these cells, you know, that are moving into one single direction, you know. It's crazy, man. It was super crazy looking at this under a microscope. Here I zoomed in, like I zoomed in a little bit to get better quality, but this is the maximum that I saw, basically, you know. I think at some point I may or may not have had like a better. Oh, is it here that I get like a better? No, I don't. I'm just gonna fast forward a little bit. Oh yeah. No, here. Yeah, because I spent like a long time trying to look for like individual ones, but here you can also see individual yeast cells or fungal cells together. And you can also see one budding in the center. If I can like, here I tried like focusing a little bit better. And this is what I wanted to showcase you about the pseudo-hyphae. They're not hyphae, they don't create a mycelium. They're a pseudo-hyph, meaning that they're not, they have that distinct shape, but they're not actually hyphae forming a mycelium. Because this is what they form. And again, you can see that they're budding from from each other, in and out. You have the water moving in, growing. And I think at this point I just kind of like, yeah, I tried finding them back again. But man, there's so many of them. This was really cool. Looking at this under a microscope. At the fungus cells. Another piece of information that I can give you guys is usually inside of like moldy cheese, what they sell as moldy cheese is you're probably going to see the Penicillium species. It's if I'm not mistaken, I think I read that somewhere. It's usually a derivative of the penicillin species, you know? That's what I kind of read, and it was really super interesting as well, because I also forgot to mention that the penicillin is also used as medication into antibacterials. Anyways, that was the infectious eukaryote ages. I'm super glad you stayed so far. If you stayed, kudos to you, because you really wanted to learn out of the fungal, about fungal species. Thank you so much for watching. I'm going to end the stream here. If you've enjoyed it, I hope it was informational. I hope it was educational. If I have any mistakes, please call me out. I will do the necessary corrections. If you've enjoyed it, please remember to hit like, subscribe, and share. Follow me for more content, and I will see you folks next time. Thank you for watching.
Hey guys, thank you very much for watching the whole content. I hope you enjoyed it. If you want to catch it live, come on twitch.tv/MajinP. That's MAJINNP. I appreciate the support this channel has been getting, which is making it growing bigger and bigger. So please take the time to like, subscribe, and share, and all of that fun stuff. I'm always here trying to get up there, so if you want to support your boy, you know what to do. Thank you very much again for watching, and peace out.